Hello, everyone, and welcome to today's webinar, From Data to Discovery, Best Practices for Proteomic Data Analysis. My name is Sydney Panilla. I am with LabRoots, and I will be your moderator for today's webinar. Today's educational web seminar is presented by LabRoots and brought to you by Seer. To learn more, visit seer.bio. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speakers for today's webinar, Serafim Batzoglou, PhD, Chief Data Officer, Seer, and Uros Kuzmanov, PhD, Senior Research Associate, University of Toronto and Ted Rogers Center for Heart Research. Now I will go ahead and hand it over to Taylor Page, who will introduce today's webinar topic. Taylor, over to you.
Thank you, Sydney. As mentioned, I am Taylor Page, and I serve as the Director of Product Management at Seer, and I'm excited to kick this off today. We're here today to share with you more about how unbiased mass spectrometry-based proteomics is really reshaping life science research and biomarker discovery. Proteomics is really entering a new era. We can now look at the proteome both at peptide level resolution and at population scale. And that really opens up vast opportunities for discovery, but it also generates an extraordinary amount of data. Today, we will share examples of how you can mine the depths of this data to reveal biological information that other methods cannot. Before we hear a few data stories from our speakers, as mentioned, I'd like to walk you through how Seer's suite of technologies makes this depth of proteomics not only possible, but practical. Originally founded in 2017, Seer is uniquely positioned to deliver on this promise of the proteome, as we have compiled a multidisciplinary team of nanoengineers, computational chemists, automation experts, and data scientists to create an innovative technology that is both robust and reliable. The science behind our technology is backed by numerous publications, accolades, awards, and IP that really highlight these innovations. We continue to expand our global footprint and access to our technology with dedicated service providers, as well as channel and technology partners to really enable the community to decode the biology of the proteome. As many of you also understand, we believe that full characterization of the proteome is really essential to understanding human biology. Almost every function within a living organism is performed by a protein or a group of proteins that interact with each other and work together. And while genomics and transcriptomics have provided a wealth of information, our collective understanding of how dynamic proteins, PTMs, protein-protein interactions contribute to higher level phenotype is far less mature. Moreover, protein characterization of the billions of documented genomic variants in the context of diverse phenotypes will require population-level, large-scale analysis to annotate their function. So technological advancements that allow us to decode the complexity of the proteome will meaningfully advance our understanding of human health and disease. There are fundamentally two distinct approaches to deep proteomics. The first is a targeted approach using antibodies or similar analyte-specific reagents. The second is an unbiased or untargeted approach using mass spec, which is considered kind of a gold standard of proteomics. If we think of the complexity of the proteome as represented by an iceberg, then the targeted approaches really are represented by just the tip. They interrogate only the specific form of the protein that their analyte-specific reagent can measure. Unbiased approaches, however, using mass spec are not constrained to measure a specific form of a protein. So when coupled with Seer's Proteograph platform, this approach provides depth, scale, and visibility into the enormous content that is yet to be discovered. Seer's Proteograph platform simplifies access to mass spec discovery proteomics at scale by consolidating the upstream proteomic sample prep onto the surface of engineered nanoparticles. That tuned surface, the chemistries, enable deep proteome profiling to comprehensively capture and to identify proteins in complex samples. Combined with turnkey liquid handling, the entire process is automated to ensure reproducibility and to minimize hands-on time. This technology is packaged into Seer's Proteograph product suite, an end-to-end workflow which enables nearly any lab to take an unbiased, deep, and scalable proteomics approach. Proteograph assay kits provide the consumables, nanoparticles, reagents, and enable end-to-end walk-away operation with the automation instrument. Ultimately, that yields digested peptides that can be analyzed with your choice of mass spec platform. The Proteograph One assay is the most recent addition to the Proteograph product suite and can deliver as much as 10 times more protein IDs than direct digest mass spec workflows. Our teams have also created an off-the-shelf data analysis software called the Proteograph Analysis Suite, or as we like to call PAS, for data processing and analysis, and that can manage the unprecedented production of data that is generated by the latest generation of mass spec instrumentation. PAS is a cloud scalable analysis platform, and importantly, it's pre-configured to work specifically for the Proteograph workflow. It allows you to quickly move from raw data to clear high-level view insights. So when a colleague asks you for a quick summary of your experiment, you can easily pull together results that are accurate, interpretable, and ready to share. Our team of data scientists are pioneers in scaling mass spec proteomics. They continually expand the field's understanding of how to manage and interpret data at this scale. And PAS makes those advancements immediately accessible to users. Before the Proteograph technology was launched, unbiased studies at scale were impractical, with the largest study limited to just 48 samples and the deepest published study reaching 5,300 proteins. Since the first shipment of the Proteograph in late 2020, capabilities have expanded rapidly. By 2022, multiple studies exceeded 1,000 samples. In 2023, depth surpassed 8,000 proteins and 15,000 sample programs began. Just this year, in 2025, a 20,000 sample population study is underway with multiple large-scale studies to follow. But the true value of the Proteograph product suite is the data it generates. With unbiased peptide-level resolution, researchers like yourselves can explore biology in ways that were not possible before. And with that, I will hand it over to Serafim Batzoglou, the Chief Data Officer here at Seer. Serafim.
All right, let me take the laptop. Thank you, Taylor. So I would like to say a few things about proteomic data analysis in general, and of course, being focused mostly on or biased towards Seer technology. So as Taylor said, our technology solves an important problem in proteomics, especially of blood samples and samples that have a high dynamic range, meaning that the most abundant proteins in a sample are much more abundant, like 8 orders of magnitude, than the least abundant proteins. So when one tries to perform mass spectrometry, which is really a gold standard technology in measuring molecules, but when it comes to plasma and other samples with high dynamic range, it suffers from the problem that it will be measuring the same molecule over and over, the most abundant ones, and for that reason, will have lower sensitivity to find the least abundant molecules that could still be very, very important biologically. So that's what our technology solves with our nanoparticle technology, basically. And other technologies as well for proteomics solve this problem in a different way. Affinity-based technologies kind of fish out the proteins using affinity reagents. We fish out molecules using our nanoparticles. And importantly, we then use mass spectrometry as our readout, which is really a gold standard for measuring molecules. So you can see here to the left what is achieved this way. So in dark blue, you see the coverage of the proteome with the latest and greatest mass spectrometer, the Astral instrument, using so-called neat digest, or basically not using our nanoparticles. So you get basically a given depth of proteins that is about like on the few hundreds or 1,000 per sample, depending on the Astral or Exploris instrument. And using our technology, you go 7 or 8 times deeper than that, and you find 7 or 8 times more proteins and more peptides per sample. To the right, you see the human proteome catalog, basically, which is a catalog of proteins that have been experimentally found to be present in various blood samples, human blood samples. And you can see visually how much deeper you go with the Seer technology rather than need direct digest. So, and especially in the lowest quadrant of the least abundant quarter of the human proteome, you find almost, a very large fraction of them using the proteograph, the Seer technology, and you find almost none if you do direct digest. Now, I think most of you are probably familiar with different proteomic technologies and have heard of SomaLogic, Olink, and perhaps others that claim to measure the proteome, and we also claim to measure the proteome. And so how do these technologies compare? One thing I want to say in a very objective way, really, is when we say measuring the proteome, it's a bit of not quite, it may be giving the false impression because each technology really measures different aspects of the proteome. First of all, each technology finds different proteins. As you see here, the Venn diagram of overlap between the 3 technologies that I mentioned. And even in the proteins that are measured in common, there are differences which I'm going to speak more about in the subsequent slides. So really, there are really 3 different, let's just say, measurements of the proteome that each are informative and quite distinct from each other. So we are really excited as Seer to see over the next couple of years, because already studies are coming out that compare between these 3 technologies. And I think we're going to learn a lot by comparing in the same samples. So at the top, you see a study that was just posted in bioRxiv by luminaries in the field, Mike Peitzner and Claudia Langenberg, on the East London Genes and Health cohort, where they measured the proteome with all these 3 technologies. And you see the Venn diagram of protein overlaps. As Claudia and Mike say in their paper, Seer measures a previously unexplored proteome. We are really excited to see what we will learn from that proteome. And in the bottom, you see a very similar Venn diagram that we produce using the internal samples that we have processed. Generally, if you do a cohort study of, let's say, 1,000 or a few thousand individuals, you expect to find more than 10,000 proteins across the cohort and more than 200,000 peptides. So we really have a very rich data type that we provide in a study. Let me speak a bit about the common types of analyses that one would want to do with proteomic data. So the idea is that you have your cohort of interest. This could be very often a case control disease cohort like cancer and healthy, or Alzheimer's disease and healthy again, or it could be just a general healthy population with many different phenotypes, or it could be there is so many variants. We are collaborating on a xenotransplantation project where basically they take clinically diseased people and plant kidneys or livers in them, and then measure the proteome over 20 or 30 days, keeping the body alive. So there's so many things that one can do, but I will talk only about the most common ones. So first, if you have your cohort of interest and it has a phenotype or many phenotypes of interest, you could ask for every phenotype, what is the differential abundance of which proteins? So basically, you ask which proteins go up or down for the people who have this phenotype with statistical significance. Or you could look for biomarkers, which is related to the first analysis, of a given disease. You can say like these proteins go up or down in a very significant way, and then you correlate them with other pieces of evidence that those are really not proxies but are involved in the disease. So you start basically looking for proteins that are important for a given disease. The downstream applications are many, including drug target identification or validation, and many other potential and really basic biological understanding of a phenotype or a disease in particular. And then one could do classification, meaning that now given new samples, can I have a statistical or machine learning classifier that tells me with a high degree of confidence whether those new samples or individuals fall into one or the other category, like fall in the disease or control category, and even more interestingly, if one could even predict whether in the future a given individual will be higher risk or lower risk for a particular disease, such as cancer, for example. Another host of analyses that are very exciting and are coming out, especially as proteomics moves to larger and larger cohorts, is proteogenomics, which basically is a fancy word for saying that one analyzes proteomic and genomic data together. One very exciting type of proteogenomic analysis is pQTL analysis. Now, let me explain this very simple term. So one wants to find genetic variants that affect the expression of a given protein. So one could have a genetic variant, a particular genomic position that has an A or a G, let's say, and those who have an A, protein X goes up. Those who have a G, protein X goes down in a given condition. That's what the pQTL is. The pQTL is the link between this A and protein X. That's it. Now, this is super informative. I don't have time to get into how one could use this, like the technical details, but suffice to say that once one has those pQTLs, then one can start triangulating and asking questions of which proteins are not just associated with a given phenotype, but which ones are causal to a given phenotype. One can ask which genes, which genetic variants are causal, which ones are downstream, which proteins basically, because if a protein is associated, let's say that people who have lung cancer tend to have this protein up. Well, is this protein causing lung cancer, or is this protein a symptom of lung cancer? That's very different, because if it causes lung cancer, then you want to use it as a drug target. If it is a symptom, then it's a different approach that one could use. So very exciting types of analysis. And as we move to large cohorts like UK Biobank and others, it's becoming more and more possible to do this analysis. So let me give a few examples of some of those types of analysis in the next few minutes. And those are examples from use of the Seer technology, and I'm sure other technologies also have their own examples. So, but we will be a bit more concentrated on Seer today. So, biomarker discovery for Alzheimer's disease. So we performed a study in collaboration with MGH on 1,800 samples, which very interestingly were collected over 12 years. So there was the opportunity to also ask questions about the progression of disease and how those are related to the proteome of the individuals. So we found 138 markers that were associated with Alzheimer's and other cognitive decline. of which, very interesting, 94 were novel. Now, this is a very well-studied set of diseases, cognitive decline and AD, and we were very, very excited to find 94 novel, and this is really a very small study, 1,800, if you think about it. 8 markers were associated with progression with this disease. So basically, individuals could be differentiated on whether over those 12 years their cognitive decline would be faster or slower, according to the level of protein expression of those 8 markers. And some of them are very exciting, and the researchers in MGH are following up on them. So small study, quite challenging to conduct because of multiple sites of collection, which leads to challenges with data, systematic errors in the data which we had to work with, but even so, very informative results. Proteomic is a company that was spun off from Seer, and it's independent for the past 5 years, focusing on cancer diagnosis. If you are familiar with Grail, Guardant, or Freenome, Proteomic is like the equivalent, but using the proteome, which they believe is much more powerful than the genome in predicting cancer. And indeed, using our technology and also multi-omic measurements of different types, they integrated a test for lung cancer, which basically provides 90% sensitivity, 89%, I'm rounding it up, 89% sensitivity with 89% specificity for predicting lung cancer. And now basically their LDT is about to come out in a couple of weeks, and they are moving forward basically with applying this to the clinic. Let me talk a bit about pQTL analysis, which was performed recently, and it's the paper is out on medRxiv. I'm sure it will be in some Nature journal soon. On the genes and health cohort, this is a cohort collected in London with very interesting genetic properties, and the team analyzing it is a collaboration between many prominent academic scientists and pharma research arms. So this is a cohort of 1,464 individuals. Using Seer technology, and it was analyzed with with many proteomic technologies, including Seer, omic, and SomaLogic. As I mentioned earlier, there were 8,000 plasma proteins that were found using Seer, including, importantly, 3,400 that were uniquely measured by Seer. And then the researchers did pQTL analysis, finding associations between genetic variants and protein levels. And they found 1,200 pQTLs. This is a quite impressive number for those who are familiar with pQTL analysis because of the very small size of the cohort. Typical pQTL analyses are done in cohorts of size 30,000, 50,000 or bigger. Importantly, over half of those were novel pQTLs found only by Seer, including a couple of hundred that were novel in proteins that are measured by SomaLogic and omic. And this is quite striking because there have been larger pQTL analyses done in UK Biobank by omic for 55,000 samples, and in Decode Genomics Iceland cohort by SomaLogic on 36,000 samples. So it's quite striking that we find in this tiny cohort so many novel pQTLs that were previously not measured, even though the proteins were measured in very large cohorts, 30 times larger cohorts. And then the researchers went on and found associations of those pQTLs with disease. Basically, the way this works as a first level is they look at the gene or the genetic locus associated with the pQTL, with the protein, and then they ask whether the same genetic locus is associated with different diseases in different cohorts. And basically, they found 690 such loci, of which then they filtered with multiple pieces of evidence from rare disease, rare variant burden to even mouse models and other. We won't have time to go through, but the paper is available. And they found basically 44, they found 21 proteins that were very confidently associated with 44 diseases using this pQTL analysis. Those are novel findings, and they are basically very interesting to follow up. So the same group has performed another analysis that they have presented so far in talks, but not yet published, but the paper is coming up, which is basically to look at the very interesting rare genetics of this cohort. This particular cohort has a high level of homozygosity, which means lots of loci in many individuals have the same copy of an allele from the paternal and maternal genomes of an individual. And as a result, there are about 100, there are 200 roughly loci where at least one technology measures where the protein is with high confidence found to be homozygous, meaning both the maternal and paternal copy, complete loss of function. So basically, we have 200 proteins in this cohort measured by some technology for which several individuals in the cohort have a complete loss of function in this protein. What does this mean? Well, one can then ask the question, okay, the protein lost its function. What about its expression in the blood? So we look at the protein expression with Seer, Olink, and SomaLogic and ask whether this loss of function causes the protein to go up or down. Now, for some proteins, you would expect it perhaps to go up. It lost its function, so it's not degrading, it accumulates, there is more of it. For some, you could expect it to go down, particularly because there are powerful mechanisms in the human biology which detect malfunctioning proteins and degrade them and digest them and kill them. So you expect to see those types of signals, and it's very interesting to basically ask the question of what happens to these proteins. So that's what the researchers did for 200 proteins, and you can see them here lined up on the x-axis. So when you see dots above the zero line, that means with color, you have statistical significance for a given protein expression to go up. And when you have them below, it goes down. So you basically, you can tell the statistical significance of those. And color-coded, you see the different technologies with Seer being green. Now, when I looked first at this slide, I'm like, okay, that's quite interesting. I don't know what it means. But then if you notice, towards the bottom of this plot, you see a bunch of greens, and you don't see any other colors. So basically, these are 23 proteins that Seer measures with very, very high confidence to have completely been eliminated in all the individuals. And those measurements, by the way, each one of them is done by 5 or more measurements, 5 or more peptides. So they are very highly confident for each protein. So the researchers really filtered for the most high confident measurements. So what you see here basically is that there are 23 proteins where Seer can very confidently say this protein is lost when it is when it is a broken copy. And it's quite interesting why you don't see the same signal with other technologies. It's something to follow up. So what I'm going to do is I would like to give more time to Uros. So I am going to skip one slide here, which would take me a few minutes to describe. I want to make a last final point here that we're really excited to move to larger and larger cohorts. Such cohorts require performing a study across, so if you want to go to 100,000 or a million size cohort, you want to perform a study across many instruments and across time. So that requires very robust data across instruments and time. So we have been simultaneously, we have developed technology both at the assay level with carefully designed controls and also at the machine learning AI level, where we basically statistically identify and correct any systematic errors. Other platforms such as SomaLogic and Olic also have developed their own technologies. So, this is something that is very important. And if you are a data scientist, I think there is a lot of fun work in this field to basically correct systematic errors across a large study. So we have been working on this as well. So I'm going to stop here and let Uros continue.
Hello, everybody. I'm trying to take control. Okay, I believe I've taken control. Does everybody see this? Hello, I'm Uros Kuzmanov. I'm a senior research associate at the University of Toronto and the Ted Rogers Center for Heart Research. I'm taking a different end. Serafim was all about big data, huge sample numbers, huge patient populations. But we at Ted Rogers chose for this one of these Insight Grant programs from Seer to actually look at the depth of data, as in using matching not only serum samples, but actual cardiac tissue samples. Now, you ask yourself why and how does the Proteograph suite apply, or the Proteograph technology, how does it apply to cardiac tissue? And it's the same issue, not to the same degree as is the case with serum and plasma, the whole dynamic range issue where the majority of the protein content within serum or plasma is these 20 proteins. A similar situation occurs in cardiac muscle tissue, whereas in the majority of the proteins, again, not to the same 99% degree or 97% degree, but to a very significant degree, the majority of the protein content can be attributed to a relatively small proportion of actual proteins expressed in the cardiac tissue. And therefore, the Proteograph suite, we had hoped, and it has actually improved the coverage within cardiac tissue. So the study that we organized, even though it doesn't include a lot of patient numbers, in this sense, we only have 20 patients. However, we have a variety of samples, both tissue samples and serum samples, as well as biological fractions of the tissue samples as well. So we have a population of patients that suffer from heart failure, and one of the ways to ameliorate this issue and help the patients is to insert a left ventricular assist device, which helps the heart pump essentially. So we have within that group of 20 patients that have had the LVAD inserted, we have had 10 patients that have then further on gone to develop right ventricular failure, which is a more severe prognosis for these patients before transplant, and then 10 that did not. So for as far as the samples go, we have had serum samples at the time of the insertion of the LVAD and at the time of the transplant. So that's 40 serum samples. And then we also had left ventricular tissue from the LVAD core that's taken out for the insertion of the device at the time of, obviously, the insertion of the device. And then we also have left ventricular and right ventricular tissue for all of these patients at the time of transplant as well. And then all of these tissues also we had biochemically split into the myofibril sort of contractile apparatus and the muscle fiber fraction and the cell lysate, which was then processed using all of the proteograph suite. So even though the actual patient numbers are small, I think we, as far as how many mass spectrometry runs, which is kind of the bottleneck, and how many samples, actual samples, peptide samples were run on the mass spec, we were at a sort of a 320 mass spectrometry runs. Plus, we also had some direct digest controls. So this population of patients and the way it was designed, it allows us for a lot of both clinically relevant and technically relevant comparisons. So obviously we can compare how the tissues and the serums behave at the 2 time points before at the time of LVAD, and then the progression to transplant. We can compare the left ventricular tissue at the time points, or at each of the time points. We can compare the right ventricular tissue at transplant time, and we can also compare the chamber compartments, and then obviously whether it's right ventricular versus left ventricular, and also we can compare the actual biochemical fractions as well. So a lot of different comparisons, as I say, both technical and clinical, that we could deduce from this small set of samples. Now here, obviously, I'm going to focus on the subsequent slides on the actual clinical questions, which can be then applied actually in the clinic down the line by identifying markers or some sort of treatment options. Is the whole question of what's the difference between the patients that do develop right ventricular failure and those that do not, which is the kind of main division. But there is also all these other comparisons which can be used. So as far as the data itself looks, even though it was smaller there, as you can see, these are the standard numbers. I would just like to point out that our numbers are a bit lower than, well, it's been some time since these were run, but our numbers are a bit lower because we use serum samples as opposed to plasma samples, which do have, the coagulation factor and associated proteins, which then does bring out the ID numbers. But we got some crazy, I don't want to say unprecedented, because these numbers have been reached in the heart tissue before, but usually through means of some sort of chemical fractionation of the peptides and multiple mass spec runs being performed on a single sample. So this is just the 2 nanoparticles and then the mass spec, a couple of mass spec runs per each sample. So these are numbers that are amenable and usable for some sort of a high throughput analysis that Serafim has mentioned, that other people have done. So we're very excited about the future prospects of that. And obviously we were limited in the patient numbers because although serum is relatively, well, it's always collected for a lot of patients, these tissues are very precious and very, very limited. They don't count in the thousands, they count in the dozens or hundreds. And we've gotten those from the UHN biobank, the Peter Munk Institute for Heart Research biobank. So these are really, really precious samples that we hope to expand on and go on further. So as far as the direct digest, so comparing it to just directly digesting and running these samples on the tissue samples on a mass spec, and then compared to the proteograph suite, and we do get a lot of, we get about a 50% increase in the number of peptides identified, which then will become, that becomes more relevant down the line when some of this variant and proteoform analysis are performed, obviously in a larger data set where it's not only 10 patients, which is kind of unapplicable at this point. And as far as the protein groups identified, we identified 30%. And I think this is pushing the limits of the actual proteins that are expressed in heart tissue. So that might be a limiting factor why we didn't get more of an increase. So to get straight into it, looking at the RV tissue at the time of transplant, if you look at the lysate and the myofibril fraction, you can see that these are what's highlighted here are the cardiac enriched proteins pulled out from the Human Protein Atlas. There's about 40 or 50 or close to 50 of them, and we see majority of them in both of the biochemical fractions, which was very useful for us if we wanted to inspect those specifically. Looking at an unsupervised analysis, considering the difference between these patient groups, these 10 and 10, the site of the major morphological and pathological difference between the patient groups, it is at the right ventricle tissue. Therefore, even in unsupervised analyses, you can see the separation of these patients based on just the most unsupervised and unbiased way, just doing a principal component analysis of the first two principal components, and you see that they're quite clearly segregate. And the outlier here that you can see, that's the same patient, which happens to have a more severe form of heart failure. And then, obviously, in a pairwise comparison, we've gotten a number of candidates using, a different strategies depending how stringent you want to be, and then we are analyzing those. As far as those significantly altered proteins, the ones that can be mapped to a specific heart cell type, and we split those, so those that can be mapped to a specific heart cell type, you can see that most of the changes are driven by cardiomyocyte specific proteins, which is expected because it's a muscle organ, and you would expect the myocytes themselves, the muscle cells, to have the highest difference, although the other ones do exhibit changes as well, and that makes sense because of in the case of fibroblasts and macrophages, you do have a lot of immune invasion as far as fibrotic events. So usually in our data pipeline, once we've done these unsupervised analyses, the different clustering, and then pairwise analysis, we usually proceed into some sort of pathway analysis. And so for pathway analysis, we use either sort of overrepresentation analysis, where you just input the gene lists of differentially expressed up or down regulated proteins, or we do some sort of a gene set enrichment analysis depending on the database and everything. And we use this to annotate the data and extract meaningful, not necessarily pathways, but even, whatever the annotations are, whether it's a subcellular component or some sort of functionally relevant issue. Next, we did the left ventricular tissue analysis again and this is for the first time we had the 2 time points. We had the ventricular tissue at time point 1, which is at the time of the LVAD implantation, and then at transplant time. As you can see here in the figure C, you can see the major difference in a sort of unsupervised principal component analysis. You see the major differences between actually which biochemical fraction was analyzed, and then again the actual time point, because these patients do progress. Whether they do develop right ventricular failure or not, they do get worse, obviously, to then get the transplant. But then when looking at the actual time points between the groups, again, there is some differences as well in an unsupervised fashion. But once you supervise it, there's quite a number of candidates, even with very stringent cutoffs. But again, this is sort of site of the disease, and you expect to see major molecular changes at this point. And again, similarly to the RV tissue, we have seen that the majority are driven by, again, cardiomyocyte specific of those proteins, significantly altered proteins that can be mapped to a specific heart cell subtype. They're driven there. So there is interest in that. And again, the pathways themselves are altered, and again, similar approach. I don't want to go into the details of this before going on to serum. So at serum, again, we had the 2 time points, and here, as far as the serum goes, using a very unsupervised analysis, we do not get the best of separations, simply considering that the majority of the proteins that are identified are not from the site of the disease, and especially from the heart. There's not so many release proteins into the circulation until very late stages of the disease. And to begin with, our patient cohort was, they all are patients that do suffer from heart failure. It's just different severities, somewhat different severities, but there are very severely affected patients. So here we also looked at some of these cardiac enriched proteins to see where we detect them as far as their rankings in the intensity. And we do get a lot of the ones that are not picked up normally in any sort of at least unbiased proteomic analysis. And as far as the actual composition of the proteins that we do identify in the serum, the majority of them do come. The ones that are tissue specific, the majority of them do come from the liver. But then again, there are the most, as far as at least the Human Protein Atlas is concerned, there are the most, the tissue specific proteins, there are most of them that are liver specific and brain specific. So heart is there, but it's to a relatively small proportion. And then to identify candidates, in addition to different kinds of criteria, we did a bunch of pairwise analysis at time point 1, at time point 2, and then looking at the overlap, and we got a certain set of candidates which we're still working out. And as far as looking at the pathways that are enriched for the serum proteins, it's interesting that the ones that are relevant for our patient group, which is obviously heart failure patients, that a lot of the pathways are associated with cardiac annotations, and as well as cytokine and chemokine cascades that are also have been strongly correlated and shown actually to be associated with the progression of heart failure. And then, of course, we did some simple, relatively pairwise, simple correlation analysis. It's one of the criteria for the selection of circulating markers that we'll see. So those are the ones that are statistically significant in both the right ventricular tissue, and we have these for left ventricular tissue from the different fractions, how they correlate with the actual serum expression changes between the two groups of patients. So we do have a set of strongly associated candidates that we can then follow up on. And as far as I know, there's been an issue of the data sharing. So for me, this is just a personal, and I'm sure all of the attendees here have seen this in a lot of papers where data is not shared. So this is my pet peeve, I guess, not so much of a pet peeve, is the actual sharing of the data. And a lot of people, they have to deposit the raw mass spectrometry data, which is for the most part, it's uninterpretable, even the way they're described. But I like a lot of supplementary tables. So all of this I like to publish, and recently I've also started sharing these all the data in a very transparent fashion using something like publicly available Shiny apps that can be interactive and whatnot. So and that's where I kind of end it. And I would like to thank the people involved here, the Gremolini lab, the Ted Rogers Center, the Bilia lab, who provided, Dr. Phyllis Bilia provided all of these tissue samples, and she has a huge biobank of cardiovascular patient samples, including tissues, serum, plasma, anything and everything. And this is a very precious resource, which we hope to proceed with in some larger studies with Seer. Thank you very much.
All right, perfect. Thank you both very much for your informative presentations. We will now go ahead and move into the Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please go ahead and do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. All right, let's go ahead and get started here. The first question that we have here is for Serafim. This one says, how important is it that proteograph data can detect splice variants and proteoforms? And what opportunities does that create for translational biology?
I think it's a very interesting aspect of our technology. So every protein is measured by multiple peptides, as many peptides as are effectively tryptically digested. What does this mean in practice? On average, we detect about 9 or 10 peptides per protein, but this varies a lot. Some proteins we detect very few because they are shorter or less tryptically digested, and some proteins we detect tens of peptides. So to detect splice variants, the robust way to do so is to have multiple individuals in a cohort and find a pattern of presence and absence of those peptides in a given protein that is prevalent across the individuals. So basically, you find that many individuals, you get detected only peptides 1 and 5 and 6 and 7, and some other individuals you detect peptides 1 and 3 and 4 and 7 and 8. And now you start saying, okay, those are two splice variants that are quite common in this cohort. So this is really exciting. The power to detect splice variants increases as the size of the cohort, as you get more and more individuals. And even in small cohorts, like 145 individuals, that was the first kind of study done by Seer on a lung cancer cohort, we found that certain splice variants associate with cancer. So you get the long form present more in healthy individuals and the short form more in cancer, things that you would not be able to see if you didn't have this type of information. So yeah, very exciting. We haven't done a lot of analysis on this, but as the cohort sizes get larger, I think researchers will find a lot of utility in this.
Perfect. Okay, it looks like this next question that we have here is for Uros. And this one says, how does the ability to correlate liquid biopsy samples with solid tissue proteomes change what's possible for biomarker discovery and clinical research?
Well, I think as I briefly mentioned, and when talking about our serum and tissue data, there is certain criteria exist for good biomarkers. One of them, and one of the major ones, or protein biomarkers, one of the major ones is tissue specificity. So you want, for the most part, this is not an absolute, but for the most part, you want the protein changes or the molecular changes that are occurring at the site of the disease. In our case, it's the heart or the liver. It doesn't matter where it is, that you have some sort of a specificity to that organ, which then you can pull out the circulation, right? So that there's criteria that they actually get secreted into the circulation early enough so we can do some sort of early diagnosis. So that's where the depth of coverage becomes extremely important. And I think as far as mass spectrometry-based approaches, or proteomics even approaches, the depth that's gotten from the Seer analysis is supremely important, which means you can see it in the tissue, see the changes in the tissue that is occurring in a temporal fashion, or even at a time slice, and then you see the concomitant changes happening in the serum. I think that's the biggest. At least that's how we look at it. So I guess that is the most important thing. And of course, you're getting those low abundance. A lot of them are not simply detectable at the tissue level or even at the serum level. So getting that depth of data is the most important.
Okay, wonderful. The next question that we have here says, how do you deal with the presence of blood contaminants such as platelets and erythrocytes in your serum samples?
Sorry, what's it?
Is this for me or?
You know what? That one did not specify. Yes. You know what? I think that one was for you, Uros. Again, it says, how do you deal with the presence of blood contaminants such as platelets and erythrocytes in your serum samples?
I don't know if this is on an informatic level or at an actual sample collection level. At the sample collection level, I mean, see the difference between serum and plasma is that plasma you collect with anticoagulants and you spin out both just the cell types. Whereas in serum, you don't use coagulants and everything coagulates, and then you spin out the coagulate and as well as all the cell types. So that's a standard procedure. On an informatic level, you can filter them out, but as far as I do not filter them out as potential contaminants, because I think the proteograph suite itself, it brings everything down to the same level, so their contribution is not as significant. So I don't do any specific steps, but I'm open to to hearing anybody else's opinion.
Perfect. Okay. The next question that we have here. So this one says, nice presentation. It's actually two questions in one. Which shiny apps are you referring that one can use to get statistical results along with tissue-specific markers?
Oh, I was more referring to how I share the data for a publication. So I'm starting it with this one, but I plan always to host, have a publicly hosted. It's just what you're looking at. You're going to get a volcano plot. You're going to get a PCA. You get some heat maps that you can select. That's more. That's what I would share. I would make of the data instead of just a bunch of supplementary figures that are often not informative depending on how they're presented. I think that's what I meant.
Okay, perfect. The second part of that question says, how can we select enrichment techniques that would work as best technique?
Enrichment techniques? I don't know what that necessarily refers to. Pathway enrichment techniques or actual... I don't know which enrichment techniques are being referred to. Can I get maybe clarification on that?
Yeah, absolutely. We can ask the submitter if they can potentially expand on that question. We'll see if we get a response here before we wrap up. If not, the questions will be tended to via the speakers. Using the registration information that was provided, the contact information that was provided at the time of registration. So if we don't have time to get to that follow-up now, we can make sure our speakers address that at a later time. Let's go ahead and wrap up with this last question here. This one is for Serafim, and it says, can you expand on the quality control pipeline? How do built-in controls and machine learning corrections ensure data consistency across large cohorts?
That's a great question. So basically, the way this is done is that in every plate, which basically is a collection of samples that are processed through the proteograph and then sent to the mass spectrometer, in every plate, we have a number of controls that we recommend using for a given study. So those come from the study itself, as well as our controls. And then when comparing data across the instruments and across time, those controls give a ground truth across each protein of basically how the level of the protein measurement systematically varies given different features such as the plate itself or the machine in which it was run or the time point. So those can then be analyzed statistically and with machine learning to give basically to measure the systematic contribution of each of those different factors. So one does basically factor analysis, if you are familiar with, and other machine learning techniques to measure the systematic contribution of those factors, and then removing that contribution to basically uncover what is the pure biological signal for each of the proteins of the proteome.
All right, very good. Thank you again, gentlemen, for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, Seer, for underwriting today's educational webinar. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. As a reminder, questions that we did not have time for and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, until next time. Thanks so much, everybody. Goodbye.
Bye, everyone.